All right, so last class we were kind of talking about blood, and we're going to finish up blood, finish up the cardiovascular system, and then dig into the respiratory system today. And we talked about blood being two parts. It was like the plasma plus what we call the formed elements. Plasma, yeah, it was mainly water. We said 92% water, 92%. It depends on your blood, who you are, your current status, around there, around 90%. What else makes up your plasma besides water?
Plasma proteins.
Yeah, so we said about 7% were these plasma proteins. And what were examples of them, and what kind of jobs were they doing?
Renin? Was renin one?
So yeah, there were lots of things. So renin actually was not a plasma. Renin is released by the kidneys to initiate the whole renin-angiotensin mechanism. But
was it the angiotensin that was in it?
Yeah, exactly. The angiotensinogen that was in there, that was part of there. We're going to see lots of plasma proteins involved in blood clotting, lots of plasma proteins involved in body defenses like the complement cascade. So they're doing lots of different functional jobs like that. What other jobs do they do?
Well, you have the globulins, which acted as carriers.
Yeah, so there was like the alpha globulins, beta globulins, which can help bind and carry molecules that don't dissolve in water well, so they can get transported in the plasma. We also talked about gamma globulins, which are the kind of unique of the plasma proteins because they're not made by the liver. Instead, they're made by lymphocytes, and those are part of your body defenses. We'll talk way more about them when we do body defenses. And what was the most common of all the plasma proteins?
Albumin.
Yeah, albumin. And again, the big part of that is to help the osmotic balance. You want to have the osmotic balance between the plasma and the surrounding cells and tissues so you don't have water moving in and out of things. So part of the plasma proteins are about maintaining osmotic balance. And then we also talked about just a bunch of dissolved stuff, right? And dissolved nutrients, wastes, gases, whatever. So lots of other stuff. So check plasma. So formed elements, this is what we're going to talk about now. This includes the platelets, which we talked about in depth last class. So I am not going to go into those in detail now with all that hemostasis. In the YouTube thing, I actually kind of cut and pasted. I put the platelet and hemostasis part that we talked about in lab, and I kind of added it onto the lecture part of the video, but it's also in the lab part still. But platelets, which aren't really cells, they're actually pinched off from those megakaryocytes. They're actually little packets of granules of, we talked about serotonin, ADP, lots of other stuff. So platelets, we've done talking about. Now we're going to talk about are the other formed elements, which are the different kinds of blood cells. So actually, let me do my type tool because that will be less. Formed elements, continued. And the most common is the red blood cell. AKA RBC, also known as the erythrocyte. Erythrocyte literally just means red cell. So red cell, the red blood cell, RBC, erythrocyte, red blood cell. They're really kind of weird atypical cells. While they are being produced in the marrow, they have lots and lots of rough endoplasmic reticulum, and they make tons and tons of this particular protein called hemoglobin. But then before they actually are totally mature, they eject their nucleus, they eject most of their organelles, and they end up looking kind of like their middle got sucked out because the nucleus is gone. This would be the side view. From the top view, they would look kind of like a disc with their cheeks sucked in. And there is somewhere around 250 million molecules of hemoglobin in here. So in one red blood cell, there's about 250 million molecules of hemoglobin. We'll talk more about hemoglobin in a second. There is no nucleus. They've ejected their nucleus. Again, because they now no longer have like the DNA blueprint for how to make new parts and pieces, they can't really repair themselves anymore. So these things basically do their job and they wear out and then they're done. They only live maximum like 3 to 4 months. And after that, they get recycled. Typically, we talked about the spleen. The spleen, sometimes people talk about as an RBC graveyard. Liver is another major place where the old red blood cells are getting taken out and recycled. The iron, which we'll talk about, the iron from the hemoglobin gets kept in your body. Other parts of that end up getting converted into something called bilirubin, which ultimately is actually kind of fun. It's what ultimately makes your poop brown is from recycling the old red blood cells. So no nucleus. There's no mitochondria in these either. So very different from a regular cell. They have very low metabolic needs. So whatever kind of energy they need, they can make just through glycolysis. And it also means since their main job is carrying oxygen, they're not going to be using up the oxygen that they're actually carrying to deliver to other tissues in your body. So their main job is carrying oxygen. But we'll see, they do other stuff too. So what we need to do now is talk about hemoglobin. So let's do that. So hemoglobin, it's a protein that got quaternary structure. There's 4 protein subunits. Actually, let me make them a different color. So there are these 4, they're called globin subunits. And these are just made out of amino acids. They're kind of typical protein. So this is basically ultimately a protein. And then there are these what are called the heme groups. The heme groups are these iron-containing molecules, so these are my heme groups. Contains iron. And each one can bind a single oxygen molecule. So if we think about one molecule of hemoglobin, this whole thing here is one molecule of hemoglobin. One molecule of hemoglobin is going to be able to bind 4 oxygens. So I always kind of think about this. If we go back here and say there are 250 million molecules of hemoglobin in one red blood cell, how many molecules of hemoglobin can a single RBC bind?
$250 million times 4.
Which is? You think about Dr. Evil?
Billion.
Yeah, 1 billion molecules of oxygen on a single red blood cell, which is pretty amazing. We'll talk much more about that when we get deeper into the respiratory system. There are different things that actually affect the affinity of hemoglobin for oxygen. So under different conditions in your body, the hemoglobin is more or less likely to let go of the oxygen and let it enter into the surrounding tissues. So we're going to revisit hemoglobin and hemoglobin's affinity. If you remember, affinity is how tightly things stay bound together. We'll be revisiting that a little later as we get deeper into the respiratory system. The other thing about hemoglobin is carbon dioxide also binds to hemoglobin. You know, you usually think about hemoglobin as binding oxygen, but it also binds carbon dioxide. So the globin subunits can bind CO2. So it's carried at a different place on the molecule than the oxygen. But it actually does affect the heme's group affinity for oxygen. So again, as we get deeper into understanding hemoglobin later, we'll see that increased levels of carbon dioxide actually decrease affinity of the heme for oxygen and make it more likely that the blood is going to release more oxygen into the tissues as more carbon dioxide builds up, for instance, which again kind of makes sense if you think about it. In general, hemoglobin is not the main way you carry carbon dioxide. Carbon dioxide we're going to see is maybe like 30% of the carbon dioxide is carried by hemoglobin. The rest is going to be carried in other ways we'll talk about later. It is the main way you carry oxygen. So oxygen is pretty much all carried by being bound to hemoglobin. So hemoglobin, main way of carrying oxygen, also one of several ways of carrying carbon dioxide. The color, the more oxygen that gets bound onto the hemoglobin, the brighter red it becomes. So, you know, bright red blood is when it's fully oxygenated. As it has less oxygen, it gets darker. It doesn't actually turn blue, but like through your skin, it looks kind of bluish. That's so people draw it as blue. But basically, the people use the color of the blood as a measure of oxygen levels or oxygenation. Like when people have low oxygen in their body, we often say they're cyanotic. Cyan means blue, so people start looking kind of bluish. You can actually, if they start looking kind of bluish, that's a sign like, whoa, they're not oxygenating as well as they should. And depending on your skin color, you know, obviously it can be hard to see if you're starting to look kind of blue if you have just more naturally darker skin, but there are different places you can look even then where you can kind of see this happening. So
does every iron have And oxygen, so is the hemoglobin, does it always have 4 oxygen?
If it's fully saturated.
Okay, but it could have less and then you'd be,
Right. So when the hemoglobin is not so bright red, it's not bound to as many oxygens.
okay.
So those little pulse oximeters. I don't know. I mean, people, those have really been in the news recently. Like people should, have a pulse oximeter around to make sure that especially if you end up getting COVID and you can't breathe well, and you want to make sure, are you like actually okay? Are you really kind of in danger? People look at what is the blood oxygenation level in your body, and you can measure it pretty straightforward. You put that little pulse oximeter on your finger, and it basically shines light and has a little detector that looks at the amount of red absorption in your blood and can determine your percent like oxygen content of your blood by just checking out the color of your blood, which you can measure just by shining light into your finger. So those pulse oximeters, they do also tell you your pulse, but they also are more importantly telling you kind of what percentage of your oxygen carrying capacity is your blood maxed out at. And normally, if your respiratory system is working well, your hemoglobin should be pretty filled up. Like as you're delivering blood to your tissues, your blood should be carrying a full load of oxygen to deliver. If your respiratory system is not working properly, and therefore you're not loading up the hemoglobin fully, that's a problem. That means the blood's going to deliver the oxygen to the tissues, but the pail is not full. So that's going to be a problem. So, in fact, today we can talk a little more about some of this, bring in the topical things about, this COVID, what is coronavirus doing? What is the COVID-19 disease? Why do people end up on respirators and things like that? That actually will play a role in today. So, hemoglobin, carrying oxygen, binding oxygen, affinity can vary by a variety of things, including how much CO2 is actually bound in here, and we'll see other things as well. So RBCs, they're like 99% of your blood cells. So almost all the blood cells are red blood cells in your blood. The other ones are much more rare, but they're important. So we're going to talk about them now. So now let's talk about white blood cells. They're also called leukocytes. So these are really different. So first, they have nuclei. Pretty much all of them, they're involved in body defenses. And they are not confined to your closed circulatory system. So if you're a red blood cell, you get created in the bone marrow, you end up in the circulation, you live for your 3 or 4 months, you get recycled in the spleen or the liver. Your entire life existed within the blood vessels. White blood cells, on the other hand, are really different. White blood cells might start in the blood vessels, but then they realize, oh, there's some action going on. The body needs me to help fight an infection. They'll grab onto the wall of the blood vessel. They'll ooze out of the blood vessel and just get into the tissue spaces. There's this word diapedesis. It's kind of a cool name. Diapedesis actually means getting and leaving the vessels just into the tissue spaces. A lot of your white blood cells are just on patrol. You have white blood cells that are just in patrol in the layers of your skin. They're hanging out and cruising through all the connective tissue. They're hopping into lymphatic vessels, moving from one lymph node to another. They're doing all sorts of, they're much more independent. Again, when we get to the immune system, we'll talk about them all in much more detail. But I usually think of these guys more as kind of this, like, you're like mercenary army in your body. And they have different levels they have different jobs, different levels of rank. Some are like commanding other ones to do different things. Some of them have particular posts. There's Kupffer cells that just live in the liver, but then other macrophages whose job is just to go everywhere and then alert lymphocytes when things get caught. So there's all sorts of activity going on. And some, even though they're kind of your cells, I kind of think about them as kind of more your allies, working independently to keep your body safe. We're all in this together, so we'll look at leukocytes in much more detail. I can give you a brief rundown of them right now, but again, we're not going to go into the detail because we'll do that when we do the immune system. So white blood cells. I'll go from like most common to least common. These are basically phagocytes. Like when you have an infection, they go off and they start eating bacteria that are multiplying in the area. And as they fill up and, kind of die, like when you have that pus at an infection, a lot of that is the dead and dying neutrophils. These white blood cells that came in, gave it their all to help kind of fight these bacteria, and now they're part of this icky pus. Lymphocytes. These are part of specific immunity. We're going to talk about lymphocytes in way more detail. I talked a little bit about that when we were talking about the idea of antibodies, how antibodies will only recognize very specific looking things. The lymphocytes are the things that make antibodies, and there's also other kinds of lymphocytes like T lymphocytes that also recognize specifically shaped things but play other roles, like around actually killing infected cells and stuff.
Do we need to, sorry.
Anya?
Do we need to know what each of these white blood cells do right now, or are you just listing them?
I'm kind of listing them.
Okay.
Yeah, for your exam, you should know that white blood cells have to do with immunity. Mm-hmm. Like if on some exam I said, like, little Billy was born without any white blood cells, like, what kind of issues is he going to have? He's going to be dead immediately of infections or something. But if he didn't have any red blood cells, he couldn't carry oxygen. So you should know it there. But in terms of the specifics here, I'm kind of introducing it to kind of prime you. But we're going to, like on the last exam 4, that's where you actually are going to really need to know about what is a B versus a T lymphocyte and all that kind of stuff. Yeah, so that's a good question. Monocytes. These actually turn into macrophages, big eaters. Eosinophils. say fight. Like worm, like if you had like, you know, some worms in your body, you would have lots more eosinophils, you know, at the moment, you know, as well as they fight other things too. Basophils, these increase inflammation. We'll talk more about what is inflammation and histamine and all that later. Basophils basically have a bunch of histamine granules that they can release and increase inflammation. Again, all of this stuff I wrote here, I did just to kind of give you a kind of laundry list of them. So when we get to them later on, they will be familiar. The little thing we use in anatomy that I was like, you know, never let monkeys eat bananas. Like if you're trying to remember what is the most to least common, you have to kind of think about a monkey. And he's like, oh, damn, I wish I could reach that banana up there, but it's out of reach because they said never let monkeys eat bananas. So that's white blood cells. What else do I want to say here? We can talk about anemia. Let's talk about a few pathologies before we leave cardiovascular. Did we mention like aneurysms and atherosclerosis yet? Yes?
Yes, we did.
Okay, so what we can do now is anemia. Anemia is when you have a decreased ability to deliver oxygen to the tissues. And there's lots of different ways you can get that. One way that you can get anemia is if you're not being able to make the hemoglobin, right? And you need iron to make hemoglobin. So you can have like iron deficiency anemia. If you don't have enough iron, you're not making hemoglobin. If you don't have hemoglobin, you're not carrying oxygen. Easy peasy. You can have hemorrhagic. This is not how you spell it, but I always forget exactly how you spell hemorrhagic. Hemorrhagic is meaning you've bled out a bunch and you don't have enough blood to actually carry all of the oxygen around. You can have pernicious anemia. So this is lack of vitamin B12. This is a vitamin that you need, among other things, to make your red blood cells. So if you don't have enough of this vitamin, you're not going to be making enough red blood cells. You can get anemia, you know, if you're undergoing radiation treatment for cancer. That radiation treatment usually takes out the rapidly dividing blood-forming cells in your marrow. They call that aplastic anemia. So that's just when, like through radiation treatment, you're killing off your bone marrow hemopoietic cells. Yeah, I don't know if I mentioned that word, hematopoiesis. This is the official word for blood forming. So hematopoietic tissue is basically the red marrow in your bones that are making the blood cells. You can have like sickle cell anemia. Sickle cell anemia is when you have one base pair in the genetic code to make hemoglobin is off. It's a genetic disease where literally just one stupid codon is off. But that is enough to make a version of hemoglobin that ends up grabbing onto itself under low oxygen conditions, and the whole blood, normally the blood cells look like these little discs, but in sickle cell, they fold in on themselves kind of like a canoe, and they stick to each other and they start getting stuck in your blood vessels and they have a hard time moving around. So there's that. So anemia is when you have trouble delivering enough oxygen to the tissues. Another thing people get is shock. Shock is just when you have inadequate blood flow to the tissues. You can have shock. You can have cardiogenic shock. Cardiogenic meaning it's happening because the heart is not working right. Like if something's affecting your cardiac output, right? If your cardiac output is plummeting, you're not delivering enough oxygen or blood to your tissues, that is a type of shock. Do I have? Hold on. Oh, there's what? Oh, hypovolemic shock. Hypovolemic shock is just, from what it sounds like, your blood volume is really low. So if you've just been bleeding out a bunch, you might have the inability to have tissue perfusion of your blood into your tissues because there's just not enough blood to push around. So there's vascular shock. If your blood vessels are got things going on, maybe they're all dilated and your pressure's run down or whatever. So shock, when people talk about shock, you might, there's obviously the shock, whoosh, I'm in shock. But when people talk about in a medical situation that people are in shock, that means that there is inadequate tissue perfusion by like oxygen carrying blood. And it can happen, again, because the heart, cardiogenic, something's wrong. The heart is not doing its job to push the blood around. It can happen because your blood volume is really low. So that word shock has a very specific meaning when we talk about cardiovascular function. Anemia is just kind of inadequate oxygen being delivered to the tissues. It can happen because your hemoglobin is compromised or things like that. So is there any other things I want to talk about? I'm only going to do this really briefly because we have so many other things to talk about, but I want to talk about fetal circulation very briefly. So here's mama. Little baby fetus is developing inside. There's like the uterus. So, the mom and the baby both have, their independent blood and circulatory systems. How is the fetal circulation going to be really different from an adult circulation?
It's going to bypass the lungs.
Exactly. So this is basically going to be bypass the pulmonary circuit. Right? Normally, you have, here's the heart, You send the blood out to the body. Come back to this side of the heart. Send the blood out to the lungs. Pick up oxygen. Come back to this side. There's no reason to send half of your cardiac output to the lungs if there's no oxygen to pick up, because you're not breathing yet. So what happens is you get a couple of these short circuits. And again, I'm not going to go in anatomy. We go way into detail and we name them. And for this class, I just want you to know in the big picture that there are going to be these bypasses. There's this bypass between the atria where blood goes from the pulmonary side, from the left atrium, from the right atrium over into the left atrium. There's actually a short circuit from the pulmonary trunk back into the aorta. Actually, where am I going? Pulmonary trunk into the aorta over here. That's not good. Geez Louise here. Pulmonary trunk to the aorta, right there. So we, like the blood that would have been going out to the lungs, it gets shunted over to this side. The blood that's returning from the body that would have gone out to the lungs is just coming back, back into this side to go down here. So bypass the pulmonary circuit, that's a big thing. Another thing that is different, where is the baby getting its nutrients and oxygen?
The umbilical cord.
The umbilical cord. So there's this whole umbilical circuit that we don't have as an adult, where you send blood out to the placenta. The mom also sends blood out to the placenta. The placenta there's no actually mixing of blood or anything, but it's a place where the fetal circulation can exchange materials with the maternal circulation. So this is a place for exchange between fetal and maternal. So the little fetus can absorb oxygen and nutrients, can give away wastes that the mother's system can pick up and get rid of. So there's that whole umbilical circuit where the fetus is getting nutrients, giving up waste, and the mothers can absorb that. So exchange, but again, no mixing. You don't actually mix the blood or anything. And then the other part, I won't even go into more detail. There's actually a bypass of the liver processing. Normally when you absorb nutrients, you actually go into that hepatic portal system, which we talked about, where you then process those nutrients. But because you're not actually eating and you don't need to process your nutrients, there's a bypass of that whole hepatic portal system as well. So that's all. I'm gonna leave it here. Just, again, in anatomy, we have more time to go into this, but a number of congenital defects happen if that transition is not made smoothly from fetal to adult circulation. If the little holes don't close properly in between the atria, or these little bypasses and shunts don't close off. So then all of a sudden your circulation is not going to be efficient because you've got mixing of oxygenated and deoxygenated blood once the system should actually be functioning as an adult. So it's worth thinking about fetal circulation, just because when things go wrong, like I said, after postnatally, that can actually lead to a number of not uncommon medical situations. Okie dokie. So any final questions, comments, or anything on the cardiovascular system? I think we're finally done with the cardiovascular system. It took just like a week longer than I thought. Okay. Now, yeah, take a deep breath and start the respiratory system. So the respiratory system is doing a variety of things, but mostly it's about getting oxygen into your blood. So then you can, the oxygen is around you in the atmosphere, and you got to get it into your blood. And then once it's in the blood, you got to get it out of the blood into the tissues. So when people think about the respiratory system, they often think about three main steps. First is called pulmonary ventilation. This is basically getting air into your lungs. The air is in the room, but you got to get it into the lungs where it can actually enter into the bloodstream. We're going to spend time talking about the mechanics of this, the pressure changes that lead to the air actually entering into your lungs from the outside. Once the air is in your lungs, you got to get it actually into the bloodstream. They call it external respiration. This is basically, so the oxygen, pulmonary ventilation was basically just trying to get the air into the lungs to start with. Then again, once it's in the lungs, you can move it from the lungs, the little alveoli, into the blood. Then there's, transport. This is the part we've just spent the whole time talking about. This is just your cardiovascular system. This is, once the oxygen's in the blood, now we can move it all over. We have 100,000 kilometers of blood vessels to get that blood to all the far reaches of your body. Remember, because we said diffusion is not so good. Diffusion is only good over a fraction of a millimeter. Diffusion will work to get the oxygen into the blood in the external respiration. But then for the long distribution, we need to do actual flow. That's this cardiovascular system. But once we get the blood at the target site where the cells are in the tissues where you want to deliver it, then we have what's called internal respiration. This is going to be, O2 from blood into tissues. Right, so we're going to talk about this as well. And I was alluding to this when I was talking about the affinity of hemoglobin for oxygen. Like, depending on the conditions in your body, you might want to deliver more or less oxygen to the tissues depending on the metabolic needs. So we'll look at things that actually affect how oxygen is released from the blood and delivered into the tissues as we go on. Obviously, once oxygen is in your tissues, you're going to have, cellular respiration. We've already talked all about that when we did our, talking about the cell. We don't need to talk about that. Instead, we're going to spend quite a bit of time looking at this. Pulmonary ventilation. How do we actually get the air into the lungs in the first place? We're also going to talk about this in quite a bit of detail. What are the conditions that affect oxygen actually entering into your blood? It's going to have everything to do with partial pressures of oxygen in your alveoli versus partial pressures of oxygen in your blood, which drive the movement of oxygen. We'll look at how that's affected by the atmospheric pressure, how it's affected by your respiratory rate, by the depth of your breathing. All these different things are going to affect how much oxygen is actually entering into your bloodstream. Transport, we do not have to talk about because we just talked about it. That's all been the cardiovascular system. We are going to talk more about this as well, the internal respiration. What kind of conditions affect your body's delivery of oxygen, how tightly the hemoglobin holds onto the oxygen versus lets go of it. So this is kind of our game plan here. And we'll start with a little bit of anatomy though. We can't actually talk about this without doing the basic anatomy. So let's start with that. Lips, these are supposed to be lips. All right. Here's our creepy looking person here. All right, so here's the tongue, nasal cavity, oral cavity, kind of just trying to giving you a sense of these different cavities here. Nasal cavity where it's just having air coming in. This is your oral cavity here where it's inside your mouth. The back of the throat, they call the pharynx. We have a nasopharynx, an oropharynx, laryngopharynx. Let's first start with the nasal cavity. One of the things when you're breathing, you want to try to filter the air. There's lots of crap in the air, dust and spores and this and that. And so we're going to talk about many levels of filtering and making sure that you are protected from dust as you breathe. The first level is actually what we call vibrissae. Vibrissae is the fancy pants name for nose hairs, right? So just big pieces of dust and lint that are floating around are actually going to get caught on these hairs that are kind of lining we call the external nares, these holes going up into your nose here. So that's the first step. The second step, the nasal cavity and then all of the lower respiratory passages are coated with this or lined with this special type of epithelium called, I'm going to write it out as we're saying. So respiratory passages and nasal cavity are lined with special epithelium. It's called pseudostratified columnar, actually ciliated pseudostratified, ciliated pseudostratified columnar epithelium, with goblet cells. What does that mean? It means you've got this columnar cells. They actually, let's not even get into the details of why it's pseudostratified. It's pseudostratified because it looks stratified, but it's not. So I'm kind of drawing this tissue right here. Cilia, those are the little things that are going back and forth, waving back and forth. Goblet cells actually make mucus. So we've got this icky gooky mucus. Mucus is this slimy, mucus. Mucus is basically snot. It's these glycoproteins that hydrate and get all kind of slippery and slimy. So this is kind of mucus. And I should say mucus, I don't have to take too much time. Mucus is another one of these things where you think we understand it better. Like I have a friend, her lab is all about just trying to understand mucus and how do the cells make mucus and how can we make effective artificial mucuses and stuff. It's not clear. Something as simple of like, oh, how do you make snot? And what is the exact composition of snot is not actually something that is totally well characterized. So it's kind of wild it's why there's lots of room if people want to study biology. There's lots of really compelling questions still that you can look at. So this mucus on these little cilia, which are wiggling back and forth. So this mucus acts kind of like fly paper. Dust comes in, it sticks, gets stuck in the mucus. And then these little cilia sweep the mucus and the mucus just gets swept to the back of the throat. If you're in the top part, the cilia sweep the mucus down. If you're in the bottom part, they sweep it up. So that's another way that you have to deal with the dust that's coming in. Dust comes in, gets stuck in the mucus. And then it gets swept to the back of the throat where you swallow it. But let me draw in a little more into my picture here just so this makes more sense. Whoops. Right, obviously your throat goes two different places. You can either like bring air down and into your lungs, or you can swallow things and it's going to go to your stomach. There's a little flapper dude here that it blocks off the trachea. It's called the epiglottis. So when you're swallowing food, this thing kind of closes down to make sure that you're not gonna get food going down towards your lungs. Obviously, sometimes it doesn't work and you choke, but most of the time it does. It works fine. It's obviously also possible to swallow, but have it go down to your lungs, like, right? Oops, I got air down in my lungs and I had to like just get it, or not air, I got air down into my stomach, right? That's air down into my stomach instead of into my lungs. So it's the same entrance into both. So as these little cilia are going, they are constantly sweeping the mucus up the trachea, if dust gets caught on the mucus down there, it's going to end up in the back of your throat and you'll swallow it. Up here, they're sweeping it downward, also to the back of the throat. So you usually are doing it unconsciously, but now that I'm mentioning it, I bet you all of a sudden realize, oh my God, I got to swallow. You constantly have to swallow. You're constantly swallowing the mucus that's building up in the back of your throat there as these cilia are working to just sweep things and get rid of any kind of dust and stuff that got stuck in the mucus to keep your respiratory passages clear. We'll talk more about this mucus. This mucus has these antimicrobial molecules called, oh my God. We'll talk about it when we get to the body defenses. But so it's not just sticky, it's also antimicrobial. So we'll talk more about that later. So you deal, and I should mention, I didn't mention the nasal cavity. It has what are called conchae or turbinates. Conchae are basically like these swirling shelves in here. So when you're breathing in, the air gets all kind of swirled around. So if you're a piece of dust getting breathed in, now all of a sudden you're getting all turbulent and up stick to the mucus. And once you're stuck in the mucus, you get swept to the back of the throat and you get swallowed and you're out of commission. Again, we're going to get to the stomach in more detail. The stomach is full of hydrochloric acid. It's down at pH 2. It's not a pleasant place to land if you're like some little spore or something that just got stuck in the mucus and now is getting swallowed and dumped down into your stomach. So vibrissae can get big pieces of lint, the big hairs. This mucus catches smaller things. Sometimes they call the mucus coming up your trachea like the mucus escalator or mucus elevator, because the mucus is constantly coming up. Part of one of the things smoking can do is paralyze these little cilia so they don't beat as efficiently, which means the mucus just kind of builds up down here and isn't getting naturally moved up to where it needs, in which case, you manually kind of just bringing it up. This whole smoker's cough is those cilia not doing their job so well to bring the mucus up, which normally is just kind of naturally always going on. So we've got mucus. Let's see, what else do we need to talk here? Let's get a little deeper into this, where this epiglottis is and the larynx here. So nasal cavity, where you breathe in. And I should, before we leave here, nasal cavity is doing a couple of things. Yes, it is helping filter the air. So this helps filter. Because you've got the nose hairs, you've got the mucus in here. It's also helping warm the air. Whatever the conditions are like outside, you're going to be bringing this air down into this really delicate tissue in your lungs. And you want to be sensitive to that lung tissue. So you want to kind of warm and humidify the air before it heads down there. We're gonna, in our lab today, part of the lab is looking at how is your exhaled air different from the air that you inhaled from the room around you. And, spoiler alert, it's probably going to be warmer and moister than the surrounding air that you breathed in. So kind of pre-moisturizing and warming that air before it gets down into your delicate lung tissue is a good thing. Mouth breathing, you're not getting that. In general, it's better, it's kind of kinder to your lungs and your throat and everything to breathe through your nose, because that does this kind of filtering, humidifying, warming of the air before it gets down deeper. So now we're going to look at this in a little more detail. This little junction here between the trachea going down to the lungs and the esophagus going down to the stomach. So let's look at what we call the larynx. This is going to be my trachea. So this is the basic idea here. You've got this junction and food is going to come down, and you want to make sure food can't get into your trachea, going to your lungs. So that's why we've got this little flappy dude, the epiglottis, which is going to flap down and block off the entrance to your trachea if you're swallowing. If you're just breathing, then the air can go in. You can actually, part of the way the epiglottis closes is the whole larynx structure slides up, which then kind of pushes up against and blocks this little entrance here. If you touch, it's worth it. Touch your, in fact, the tip of the front of your larynx, this is your Adam's apple. The Adam's apple is basically the laryngeal, the cartilage there on your larynx. And if you swallow, you'll feel it slide up. And that is actually helping block off the epiglottis as you swallow so the food doesn't go down into your lungs. And then when you're done swallowing, you'll just feel it slide back down. So that thing of your larynx sliding up and dropping back down when you swallow, that's about blocking off the epiglottis to make sure that you don't choke, basically. The other thing that is in the larynx that we should talk about briefly are the vocal cords. So I'm going to talk about them briefly. So let's look at a cross section. If I did like a cut through here, so I'm kind of just looking down to see what do the vocal cords look like from the top. So this is like looking down larynx. And here are my vocal cords. Vocal cords, also known as vocal folds, they're basically these little membrane things that are anchored on these cartilages. This is kind of called the arytenoid cartilage. And you have little muscles where you can swing these back and forth, open and close the little gap between them. So this gap here is called the glottis. So one thing you can do with your vocal cords actually is close down or open up the passage of air going down into your trachea, right? So if I shut down the glottis here, then air can't move up and down. Like, so if you're like bearing down for a bowel movement, like you want to like compress all this stuff, but you don't want the air going out, you shut down the glottis. Next lab, we're going to look at coughing. If you want to cough, build up a lot of pressure and then open the glottis and let the air escape in one big impulsive blast, right? So the glottis is this way of shutting down the flow through the larynx here. Open or close those little vocal folds. So that's one thing you can do with the vocal cords. That's not about talking, but that's about making the air passage open or closed. The other thing, obviously, is they vibrate as air flows by them. And you have other muscles that actually work. They actually tilt your larynx back and forth and change the tension on the vocal cords. And depending on how taut or relaxed they are, they're going to vibrate at higher frequencies or lower frequencies. Like, you know, tighter, taut, more tautness, kind of as showing you with the guitar strings when we were doing the cochlea and the basilar membrane. Something that's more taut is going to vibrate at a faster, higher frequency. Things that's more floppy are going to be vibrated at a lower frequency. So by altering the tension on the vocal cords, you change the frequency they vibrate. That's the only thing your vocal cords do is change frequency. They vibrate and make kind of a frequency buzz. I can go, so that's what they do. Everything else about your voice is kind of post-processing of that basic buzz using your mouth and your tongue and your lips and all that. Like I can have like a basic buzz. That's just changing the resonating chamber in my mouth. Like that's just changing the shape of your mouth and your tongue. Like plosives, like puh and tuh, you know, you could use your mouth and your tongue to make those kind of things. There's fricatives, you know, but that's not the vocal cords. The vocal cords just make a buzz. So let's just, so vocal cords buzzing at different frequencies. And part of post-puberty sexual changes, like as like testosterone goes up in guys, the larynx enlarges. That's why guys have more protruding Adam's apple, because that larynx is getting bigger and that, like they call the thyroid cartilage, is getting larger and you can see it poking out on the front of their neck. Also, the vocal cords get kind of larger and thicker in that process, which is why the guy's voice is in general lower than a woman's voice, just because that kind of larger, heavier vocal cord is going to vibrate at a lower baseline frequency. So that is vocal cords, and that is the larynx. So we have the larynx. Again, we have the esophagus, but we're going to talk about the esophagus later when we do digestive. We have the trachea here coming down. We do actually, let's leave the esophagus here for one moment. Most of your tubes can stay open just because there's stuff inside. You know, your esophagus, there's going to be a bolus of food pushing. In your blood vessels, Your blood vessels stay open because the blood has pressure pushing and keeping them open. For all of your air passages, they need some kind of stiffening to keep the walls of the tube opening, or else they'll collapse in, and then you're not going to be able to get air moving down. So people talk about keeping airways patent. It looks like patent. If patent is the way you pronounce it, it means keeping them open. So especially all of the larger airways have reinforcing cartilage to uphold them open. So in your trachea, you have these like cartilage rings that are going around. That help keep this thing open so it doesn't collapse. If I do a cross section through it, they're actually not totally rings. So if I look at my cross section there, here's my trachea. The cartilage only goes part way around, kind of like a C shape. And then you have the esophagus behind it. This way, when you're swallowing food, let's say you've got some bolus of food going down your throat, it's not going to go whack, 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 whack on all the little rings as the food goes down. So the trachea has these cartilage rings to keep it patent, but they have little gaps on the back where the esophagus can expand. Once you get further down into what we call the bronchi, then the rings actually go all the way around because there's no longer any worry about the bolus whacking on anything. All right, so what we're gonna do is look at our respiratory tree is coming down. The respiratory tree is like your trachea. And then it starts branching. So this is going to be what we're going to call the conducting zone. Basically, the trachea branches to the right and left. They're called the primary bronchi. And then those continue to branch into the secondary bronchi, the lobar bronchi, going into the lung lobes. There are actually only 2 lobes on the left because your heart's taken up more space on that side, 3 lobes on the right. They branch even further into what are called, the third level of branching is called the segmental bronchi. That's actually a clinically useful thing to know about. Each of these segmental bronchi enter an area which is called a bronchopulmonary segment. And those tend to be kind of walled off from each other by connective tissue. So if there is some kind of infection in the lung, sometimes it's confined to one bronchopulmonary segment. So these are kind of little sequestered areas. But then it keeps branching. There's going to be 23 orders of branching. It's going to go more and more and more and more into tinier and tinier and tinier and tinier little branches. Again, this is all conducting zone. It's just bringing the air down to where you're going to have gas exchange. There's no gas exchange happening. It's just bringing air down. As we get into the smaller ones, they're called bronchioles. These no longer have cartilage supporting their walls. They're small enough that just the smooth muscle in the wall is got enough kind of stiffness to keep things open. So these no longer have cartilage. what I can't spell muscle, just smooth muscle. It's important to kind of think about the bronchioles with their smooth muscle. These are activated by the parasympathetic and sympathetic nervous systems. Sympathetic will dilate these bronchioles, make it easier for airflow to go down. Remember, flow of air is got the same basic mathematical rules describing it as flow of blood. If you dilate bronchioles, less resistance to flow, that means it's easier to get the air down. If you constrict them, that's going to be the parasympathetic system. Parasympathetic activation constricts bronchioles, increases resistance to flow, which means it's going to be harder to get the air down into the deeper parts of the lung, or for that matter, up out of the lung. In asthma, for instance, asthma is basically an allergic reaction that constricts the smooth muscle in these bronchioles, and that is going to increase resistance to flow, is going to make it harder to breathe. You got to create much larger pressure differences to drive the flow of air down into your lungs. And conversely, part of asthma is not just getting air down into your lungs, it makes it harder to get the air out of your lungs, because the resistance to flow doesn't care whether the air is coming in or out. So all of this branching is going to finally bring us down to the place where we're going to finally actually have gas exchange happening, which are going to be the little alveoli. So at the end of all of this branching are going to be our alveolar sacs. So that's at the end of 23 orders of branching. These are going to have where the capillaries are going to come to pick up. It's actually going to deoxygenated blood's going to come in, oxygenated blood's going to leave. This is where oxygen actually goes from lung tissue into the bloodstream. Because there is so much branching, you've got a huge surface area of alveoli. People talk like the size of a racquetball court. So a huge surface area, even though it's all compacted into the lung tissue in your chest, just because there's so much crazy branching. So what we're going to do now is focus what's going on here in the alveoli and the alveolar sacs, where the actual gas exchange between the blood and the lungs are happening. This zone down here, this is what we're going to call the respiratory zone, where the actual gas exchange is happening. Conducting zone, all of this branching is all part of the getting the air down to these dudes. I should mention, this conducting zone is sometimes kind of called the dead zone or dead air. If you think about it, when we're ventilating our lungs, here's let's say this is our respiratory zone at the very bottom. Every time you breathe out, you might be breathing out a lot of the air that's in your alveoli, but this stuff is all just open piping. So this is, even when you're done with your exhale, there's still air lingering in here. This is like our dead zone here. And then now this is kind of the stale air that you were not able to breathe out. So now when you're breathing in on your next breath, that is going to just mix already in with the stale air that's already in here. And therefore, the air that gets actually down into your alveoli, when we look at it, is not going to have as much oxygen as the air that you're actually breathing in from the outside, because the air that ends up down in your alveoli is always going to be mixing with this stale air that's already in this kind of dead space here. You know, and the more dead space you have, the more this happens. This will come up actually in the spirometry lab on Wednesday. We'll revisit this. So let's look at what's happening down in the respiratory zone. All right, so you've got, we call a terminal bronchiole. That's basically the end of the line for these little, that 23 orders of branching. And then the alveoli, each alveolus is basically like a little grape, and an alveolar sac is like a cluster of grapes. So this is called an alveolus. This whole thing is like the alveolar sac. Each alveolus, there are 3 kinds of cells that we find down in here, and we need to look at each of them individually right now. The walls of the alveoli are made out of simple squamous epithelium, this kind of thinnest kind of tissue you can get. Right, and on the other side of that are going to be your capillaries. And we've learned that capillaries, the endothelium capillaries are also made out of simple squamous epithelium. And in fact, they actually share a basement membrane for this, the thinnest possible diffusion distance. So here's our like red blood cell here. There's our RBC sitting in the capillary. This is going to be this really minimal distance that oxygen is going to have to diffuse to get from inside the alveolus to get into the bloodstream and get absorbed, get attached to the hemoglobin here. And the blood slows down here. It takes, I think the blood spends about, I don't know, like a third of a second in moving through here, which is plenty of time to equilibrate. Basically, the oxygen is going to equilibrate between the alveolus and the RBC. We're going to talk about that process in much more detail in a little bit. It's all going to depend on partial pressures of oxygen. Right, oxygen is going to move based on pressure, just like water, you know, blood moved by pressure. Other cells that are important here, surfactant cells. These are more kind of cuboidal looking cells rather than squamous. So surfactant cells. So let's talk about, so normally, this is kind of interesting, even though we're kind of low on time in some ways, and normally I would kind of move past this faster. I do want to spend a little more time here because these are the cells that get taken out by the coronavirus and cause the COVID-19 disease. So these are at the core, like why people end up on respirators and even die, is having these surfactant cells affected by this virus. So let's first talk about why we need them. So all of this area in here is moist, covered with water. So if we, let's put just a lining of, this is just water. water is lining the inside of all these surfaces here. Right, and H2O, those are polar molecules and they have attraction to each other. So if we think about an H2O and another H2O, right? Oh, yeah, let me draw it. Dagnabbit. If you remember the hydrogen bonding, wherever like the more negative oxygen gets attracted to the more positive hydrogen. And so you, again and again, you get all of these kind of attractions between different water molecules, and it creates this attraction between them within the liquid drop. But if we're at the surface of water, you have them particularly strong, all grabbing onto each other, and we call that surface tension. And I think I can do this. I want to actually, just to make it clear how powerful this actually is, I'm going to try to show this to you. Hold on, I got to turn off my background screen here. So what I'm going to do is, let me actually dagnabbit. This will work. All right. Here's a bowl.
Can you put your screen to your camera? It's on the.
Oh my God. Thank you for telling me. All right, so here's a little bowl. I have water. This is just plain water. I just kind of, during the break, I kind of got my props together here. So there is just water in a bowl. And now, where's my other props? Back. Hold on, I actually. Here they are. My other props are, prettier here, a razor blade, which is made out of steel. This is a regular razor blade. I don't want you to think it's not a real razor blade. It's like, it's cutting, it's steel. It's actually pretty heavy. This is water here. You can kind of see it wiggling. I'm using this little paper clip as, ow, fuck it up. I'm going to edit out that little, what I just said there. Dark dagnabbit. I didn't mean to just drop it there. So I'm going to take my razor blade and very gently lower it onto the surface of the water. And I think you can all see that. Maybe I can even. Can you all see that it's just floating there? Razor blade, it's floating on the water. Do y'all see that? I can like.
Yeah.
So that is the surface tension that is causing it to float. So that surface tension is really strong. That surface tension is strong enough to actually keep you from being able to inflate your alveoli, because it's pulling the diameter of the alveoli close together. So how can I break this? In fact, do you see the water actually dipping down where the razor blade has gravity pulling it down?
Yeah.
So how can I break that? How can I break that surface tension?
Force or pressure.
What kind of things do we have to, so I need something that's going to go and disrupt the attraction between the water molecules. right?
Soap?
Soap. So here, I've got soap. I'm getting some soap. This is just like dishwashing detergent. And I'm going to put a little bit on toothpick here. So I got some soap. This is basically like kind of like phospholipids, amphipathic. It's basically going to have one side that is polar, one side nonpolar. So the polar side will grab onto the water. The nonpolar side is then going to not attract anything. So each water molecule is effectively isolated from its partners rather than pulling on each other. So let's see what happens if I put some detergent here in with my, did you all see that? Did you all see the razor blade just all of a sudden just fall to the bottom?
Yeah.
So that is surfactant, is basically detergent. So I'm going to get back to where we were now and keep talking about things. I just spilled water over everything, but it's all in the name of science. So let's get back. I didn't mean that. Let's see. How about that one? All right. So let's now continue with our discussion of all this. So again, we're in our alveoli. Again, the main walls of the alveoli are these simple squamous cells. Which are super thin to reduce the distance oxygen has to diffuse to get into your capillaries. Again, you got your blood cells on the other side where the oxygen is going to be able to move into, get absorbed into your bloodstream. But then, like I said, there is water coating all inside here. And that force, which was strong enough to float that pretty heavy steel razor blade in the macroscopic world that I just showed you, all those water molecules are pulling on each other, which is basically making, when you want to inflate the alveoli, all the water molecules are pulling and going the opposite kind of force. So luckily what we all have are these little surfactant cells. And those little surfactant cells make surfactant. Oops. Right. And in fact, when babies are born prematurely, you also have to put them on ventilators because their surfactant cells are not working yet. And it takes a lot more force to try to get their lungs to inflate. Again, part of the problem with this COVID-19 disease, it attacks the surfactant cells, and which means you're not breaking up that surface tension, which means it's going to be way harder to try to inflate your lungs because you're having to fight the surface tension, which is working to collapse down the alveoli. So just kind of, yeah, again, I think normally I wouldn't have put as much energy into that, but I think it's useful to really think about how important these things are and how strong and powerful that surface tension is. You know, again, we saw it's floating a piece of steel. So surfactant cells, again, you have those cells that make up the walls of the alveoli. And then the other cells that we find down here are called the alveolar macrophages, also called dust cells. I kind of like to call them dust cells because that's kind of a cool name. They're basically macrophages. They're just on patrol, patrolling through your alveoli. And they basically eat stuff. So even if tiny little bits of whatever do get all the way down there, you have these macrophages, big eater white cells that can eat things and take them up and move them off to your mediastinal lymph nodes or whatever. So, you know, all these levels of protecting you from getting dust down into your lung tissue. We started with the nose hairs, the vibrissae, and then the mucus lining your respiratory passages. We have dust cells all the way down at the level of the alveoli themselves. There's actually one more thing. You have those explosive air blasts, right? If you have stuff, achoo, sneezes and coughs, those are ways to clear stuff out. You know, what's the difference between a sneeze and a cough? Right? Anybody? What is it? What it, you know, a cough, your explosive blast of air. What's a sneeze?
It's out of your nasal cavity.
Right, it's out your nasal. So one of them is an explosive blast of air that's trying to clear more of your upper respiratory passages. A cough is more a explosive blast of air that's working more to try to clear out your lower respiratory passages. But they're the same basic idea. You close down the glottis, build up a lot of pressure down here, and then let it all go. Really, it's a very high pressure difference. So you got this blast coming through. So now we've gotten into kind of the anatomy and a little bit of the physiology down here of like why we need surfactant cells. So what we're going to do now is focus on, let me see, actually now we can get into those three different things I was talking about. One was pulmonary ventilation. How do we actually end up getting the air down here in the first place? Then we were talking about that external respiration. How do we get the oxygen from the alveoli into the blood cells or into the bloodstream? And then the last piece we'll have to talk about is how the oxygen is released from the blood into the tissues. So let's start with number one here. How do we actually get the air down into the lungs themselves? So let's do that. Pulmonary ventilation. Pulmonary ventilation, bringing the air that's in the room down into your alveoli and inflating your alveoli. We need a little more anatomy for this again. Again, I'm going to simplify things though. So here's your thoracic cavity. What defines the boundary of your thoracic cavity and your abdominal cavity?
Your diaphragm?
Your diaphragm, exactly. So the diaphragm, it's a muscle, and it's a little non-intuitive. When it's relaxed, When the diaphragm is relaxed, it looks kind of more like a dome. When it contracts, when it pulls and tightens up, it goes down. It kind of flattens out. So when it contracts, it's going to go down and actually increase the volume of the thoracic cavity. When it's relaxed, it kind of ends up coming up more, and we have a smaller volume of our thoracic cavity. Now we need to add in, here's our like trachea and our bronchi, and let's put in our lungs now. These are supposed to be your lungs. Lunges. Here's lungs. Now we need to add one more thing. So we've talked about the idea of serous membranes, how you, like the heart, remember the pericardium? There was one layer of this slippery membrane on the surface of the heart and another layer of this slippery membrane on the body wall. Again, serous membrane. In just quick review, you have the organ, whatever, whether it's your heart or whether it's your lungs or whether it's your digestive organs that we'll see later. You have a visceral layer, the layer of the serous, again, we're talking about serous membranes here. The visceral layer is the one across the organ itself. There's going to be a second layer, it's called the parietal layer. Which is a layer that's more towards the body wall. And then there's this slippery fluid that is in between. They call the serous fluid. So that means that this inside thing can move without rubbing against the stuff outside, because there's this slippage between the visceral and parietal layers. And they slip, but they don't pull apart. There's a suction. So it's important that there is a suction between these two. Again, if you've ever had, like you're washing microscope slides, if they're wet, you can slide them back and forth, but if you try to pull them apart, they don't come apart. There's a suction. So if you pull, if I pull on this parietal layer, it's actually gonna pull the visceral layer because of that suction, assuming that there isn't like a hole that's breaking the suction. So that is gonna be critical in understanding how we breathe. So again, we're gonna have like kind of the body wall. So here's my thoracic cavity. Got my trachea, my bronchi, got my lungs. But then we have these serous membranes. So with the lungs, it's called the pleura. This is going to be my parietal pleura, not my visceral pleura. That's pleura is just the serous membrane around the lungs. There's going to be the parietal pleura, the layer that's on the wall of my thoracic cavity. And what's going to be super important is that there is a suction between these. So what that means is if I change the volume of my thoracic cavity, if I expand my thoracic cavity, because there's this suction between these two layers, it's going to also expand the volume of the lungs. So this is going to be a critical part of how pulmonary ventilation works. You know, the lungs don't have muscles, and the lungs can't really do anything on their own. But your thoracic cavity sure has muscles. It's got the diaphragm underneath. You got intercostal muscles, can change the size of your rib cage, the scalenes, and you've got a lot of variety of muscles that can actually change the volume of your thoracic cavity. And as we change the volume of the thoracic cavity, that is going to be able to actually change the volume of the lungs because of this suction between the pleura. So let's see how this all works. I should mention one more thing. Back when we look at lung tissue itself, lung tissue, we talked about surfactant because of the surface tension. So the residual surface tension is naturally going to want to make the lungs collapse in. If you just left to themselves, these little balloons want to get smaller because water tension pulls in. And then there are also elastic fibers that are kind of all around in the lung tissue. Which makes the lung tissue stretchy. So this is actually an important part of the respiratory physiology, is understanding that lung tissue is naturally kind of stretchy. And if you kind of inflate it, it's going to naturally want to collapse down. The amount of stretchiness that the lung tissue has is called compliance. So normally when you inflate your lungs, we're going to see when you want to exhale, it's pretty much passive. The lungs naturally are trying to get smaller and get the air out. This can get compromised in like emphysema, where the lung loses its stretchiness and makes it actually a lot trickier just to exhale even. So let's get back now to our picture here. So there's I'm going to first say it in words, and then I will kind of write it out in letters. The basic idea is it's all going to start by increasing the volume of your thoracic cavity, which we can do with muscles, mainly by contracting the diaphragm, which is going to increase the volume here. And because of this suction, it's going to increase the volume of the lungs. Now what we need to do is think about, well, how does volume affect pressure? And that's going to take us to the famous Boyle's law. Which basically says volume and pressure are inversely proportional to each other. If volume goes up, pressure goes down. If volume goes down, pressure goes up. Again, it makes sense even just intuitively. If you think about pressure, pressure is just force per area. And an ideal gas you can think of as little molecules floating around, and every time they hit the wall of like some box they're in, they're going to exert some force and there's some pressure. If I have a much larger box with the same amount of gas, they spend a lot more time moving through space, a lot less time whacking on the walls, creating some pressure. If I take that same gas and then I decrease the volume, so now they're all confined in a much smaller place, now they're hitting the walls much more, and there's much more of a force against the wall. So Boyle's law is pretty intuitive. The larger volume you give the gas to just float around in, the less overall pressure, the less time it's going to be hitting the wall and exerting force. So this is critical for understanding our pulmonary ventilation. So now we can put that together with this, increasing the volume of the thoracic cavity It's going to increase the volume of the lungs because of this suction, and increasing the volume of the lungs is going to create a drop in pressure in the lungs because of Boyle's law. If volume is going up, pressure is going to be going down. So I am going to, in the interest of quickness, I'm going to kind of type this out. So the basic thing that's going on here, the first step is you're going to have muscles contract in that are going to increase the volume of your thoracic cavity. So diaphragm and other muscles contract that increase volume of thoracic cavity. So the diaphragm is the main one but there are, again, the intercostal muscle scalenes and things, especially if you're doing forced inspiration, you're probably going to have other muscles, even your, back muscle, you're kind of increasing, there's lots of ways to increase the thoracic cavity volume, but in general, the diaphragm is the main one involved. And 2, because of that pleural suction, this is going to increase the volume of your lungs. Right, so if you did not have that suction between the pleura, the lungs wouldn't be pulled along. Now, this is where we get to Boyle's law. As lung volume goes up, pressure is going to decrease. So now we have low pressure zone in the lungs, and it's still regular pressure outside. So just like blood flow is driven by pressure differences, airflow is driven by pressure differences. So this pressure difference is going to cause the air to flow from outside from the room into your lungs. Twitch. So one of the things that's really important to pay attention to here is that this is so different than blowing up a balloon. When you blow up a balloon, you are pushing the air into the balloon, which increases the pressure in the balloon, which then under that higher pressure causes the walls to expand and it gets bigger. Here, some animals, like a frog, does that. A frog is doing what's called positive pressure breathing. It goes kind of forcing air down into its lung, and then that causes the lungs to inflate. But we're doing something very different. It's called negative pressure breathing. We actually expand the volume first. Volume changes, which causes a low pressure zone. Then the air rushes in to fill in that low pressure zone. So like the thing I use, sometimes I show, like I can do, like, when you learn, if you can do this, this is what I'm doing in my lungs. If I use my abdominal muscles to change the volume of my stomach, I can go, right? All I was doing there was using abdominal muscles to change the volume of my stomach and creating low pressure zones in my stomach, and air rushed into my stomach, and I got to do that fun burping stuff. But that's what's going on. You're basically expanding the volume, making a low pressure zone, and then the air rushes in. To exhale, it's gonna be kind of the opposite. So expiration, this is normally more passive. Basically, diaphragm, etc., relaxes, and volume of thoracic cavity goes down. When the diaphragm or when the thoracic cavity is no longer pulling with that suction to keep the lungs at a larger volume, the lungs naturally want to be smaller, so the lung volume is going to go down. And now we're back to Boyle's law, right? As the lung volume goes down, that means the pressure in the lungs goes up. And as high pressure lungs, in the high pressure in the lungs compared to the outside means air flows out. Again, note that this is more passive because you're basically just relaxing the muscles, and the lungs naturally, because of their compliance or stretchiness, naturally want to be smaller anyway. So if you are normally just breathing in and out, the inspiration is going to require muscle contraction and some energy, but all you got to do is just relax, and the exhale will kind of happen on its own. So this is the basic idea of pulmonary ventilation. This is inspiration for the top part here. Really important, like I promise you on your exam, I'll ask you questions explaining how this works. You really need to remember the causality. What causes what here? Like, it's not like air went into your lungs and they got bigger. It's the opposite. The first thing that happens is, lung volume increases. Then the air flows. The air does not flow. This happens first. The air flowing into the lungs happens second. So that's really important. You're going to lose points on your exam if you tell me, oh, the air flows into the lungs and then the lung gets bigger. That's not what happens. The lungs get bigger because of the diaphragm and other muscles expanding the thoracic cavity, which then pulls and expands the lungs because of that suction. And then that increase in volume lowers the pressure. That low pressure zone causes the flow of air to come in. So does that all make sense?
Yes.
Okay, so then what happens? You go back to my picture here. What happens if you get shot or stabbed? This happens. People get shot or stabbed in their chest. It's like, boof, ow, oh, I'm bleeding. Let's get a knife. Boof, there's some dagger. We got some hole right here. What is going to happen if we have a hole in the wall of the chest cavity here?
The lungs are going to collapse because you can't create low pressure within them.
Right. Remember the lungs are staying their size because of this suction between the pleura, visceral and parietal pleura. If I break that suction by putting a hole in the wall here, then that suction is not going to stay there anymore. The air can just rush in from outside, and the lung naturally wants to be smaller. We talked about that compliance. So the lung gets smaller. So now we have a collapsed lung. And we have just a bunch of air sitting in the space in here. So, and it doesn't matter how much you move your chest cavity in and out here, it's not going to change the volume of the lung because there's no suction. That suction is what couples the lung to the lungs volume to the thoracic cavity volume. The official word, there's two words we can use here. The official word for getting air into that space is pneumothorax. So pneumo, air, thorax. So pneumothorax is giving you, it's got air in there, and the actual word for a collapsed lung is atelectasis. That actually is the official word for a collapsed lung. And again, as long as that hole is there, you cannot couple, there's no coupling between changing the volume of your thorax with the volume of your lung. Luckily, the two pleuras are independent, so you can actually collapse the lung on one side and still have the other one working. So it's not necessarily catastrophic totally if you have a collapsed lung here. How are you going to deal with this? What do they do in a emergency setting if someone has a collapsed lung?
An occlusive dressing.
So exactly, so you're going to have to seal up the hole. And then what's the next thing you have to do? There's one other part to it. You're going to have to drain that air out and restore the suction. So you need to do some kind of a dressing that is going to block the hole. And then you also have to have some kind of one-way valve that gets the air out and restores the suction. So you kind of recouple the movement of your chest cavity to the volume of your lungs. So, yeah. You know, and there are other ways. You can get pneumothoraxes from a perforation of the chest. You can also get pneumothorax if there's some kind of a rip that happens on the lung side as well. You can have air get into that space from the visceral side. But you know, my description here has been like if you got like stabbed in the chest or shot in the chest or something. So that is pulmonary ventilation. Let's see, let's talk about now what happens now that the air is in the lungs and we got to get it into the blood. So we need to do a little bit of your old chemistry stuff for this. So I am going to talk about a little bit about partial pressure. So air. The air in the room around you, what's the main gas in the air around you? Oh, come on. What's most of this? Over, you know, almost 80% of the air all around you is which gas?
CO2, isn't it?
Actually, no. It's only a fraction, tiny fraction of a percent CO2 in the air around us.
Nitrogen.
Nitrogen. It's like around 80% nitrogen. And the rest of it is mostly oxygen. So it's like 21% oxygen. So obviously I said, so a little less than 80% nitrogen. And so 21% oxygen. So, and the oxygen is what we're concerned with right now. the driving force for gas, as it was for blood, is pressure differences. When you're dealing with a gas that has many constituent parts, a mixed gas like air, you can follow the movement due to pressure differences just by looking at the partial pressures of the particular gas you're interested in. So I don't know if people remember Dalton's law of partial pressures. It's basically saying that the pressure, the partial pressure of a particular gas is just going to be the percent of the overall pressure of the gas, depending on the ratio of that gas in the mix. So if I look at normal gas, like if I'm at sea level, normal air is around 760 millimeters of mercury pressure. This is just, you know, air pressure at sea level. But because oxygen is only 21% of that air, then the actual partial pressure, the pressure due to O2, is only going to be 21% of that. So it's going to be 159 millimeters of mercury. So again, that is just going to be, right, 21% of the overall pressure. So this is the partial pressure of oxygen in the air when you are at sea level. And now we get down into the alveoli. We talked about all that dead space. That dead space means that that air that's coming down into your alveoli is actually mixing with stale air that's already in that dead space. So when you get down into the alveoli, it's actually not going to be that high. The partial pressure of oxygen down in the alveoli is somewhere around 104 millimeters of mercury. Again, that's because you're mixing stale air with fresh air every time you breathe in. And then we can think about the capillaries coming down on either side. And our blood cells. So blood returning from the tissues has a partial pressure only of about 40 millimeters of mercury oxygen. So the blood cells are coming in with about 40 millimeters of mercury pressure of oxygen after they've delivered their load. Now they're coming into the zone of the alveoli where the partial pressure of oxygen in here is 104. Which way is the oxygen going to move?
From high to low.
Exactly. The oxygen is going to move into the blood until it equilibrates. Oxygen will move until by the time the blood is leaving this area going on to the next place, which is going to be back to the heart to deliver the blood, we're leaving at about 100 millimeters of mercury partial pressure of oxygen. It's basically equilibrated with the alveoli. So the blood comes in with low oxygen, leaves at about 100 millimeters of mercury of oxygen partial pressure. We'll come back, revisit this when we talk about the delivery of oxygen in the tissues, which I'll do that on Wednesday. But so don't forget this. Remember that as the blood is leaving the lung tissue, it's got about the partial pressure of oxygen on it's about 100 milliliters of mercury. That's going to make a difference when we look at, does the blood want to hold on to the oxygen or does the oxygen want to flow off? Carbon dioxide is the opposite. I'll do this really briefly. You know, for CO2, in fact, I'll just type it in just to make life easier. So carbon dioxide is actually higher in the returning blood than in the alveoli. So the carbon dioxide goes the other way. So the pressure, the carbon dioxide is way more carbon dioxide inside your lung tissue than outside in the outside air. You know, carbon dioxide, because remember, every time you breathe in and out, you are not totally breathing. In fact, you're not totally collapsing your alveoli, plus you have all that dead space. So you're always having stale air inside your lungs. So this is the basic idea here. And what I want to do now, I want to show you something really kind of a really fun little app that I have. Let me look at yeah, we're actually totally good here. All right, so this basic idea, we talked about outside, you have a partial pressure of oxygen of 169 millimeters of mercury. But inside the alveoli, you've only got 104 millimeters of mercury. So that's because, you know, you're always mixing stale air with the fresh air that you're bringing in. You know, I could increase this by bringing in, like, you know, kind of bringing deeper breaths or breathing faster. And the same thing, CO2, the CO2 that's building up here, if I am ventilating more, I can have less CO2 building up in here. So it's really kind of fun to see how breathing deeper or faster changes the actual partial pressure of the oxygen and carbon dioxide down in your lungs. Also, altitude. You know, this here I was saying our partial pressure of oxygen is going to be at sea level. You know, that's sea level, but when you're at different levels, different altitudes, if I have it here, like Lake Tahoe, which is 6,200 feet, the outside air pressure is only 609 millimeters of mercury. If you're on the top of Mount Whitney, the tallest peak in California, I think in the continental US, pressure of oxygen outside is only 429 millimeters of mercury. If you're on top of Mount Everest, which is at 29,000 feet, the pressure of oxygen outside is only 226 millimeters of mercury. So what that means is this is going to depend on your elevation. And that also means then as we go into the lungs, when we get in the lungs, this value here is going to be affected by your elevation because that's affected by what you're breathing in. So what I'm going to do now, this is going to take me just a moment to try to make this work. I'm going to try to sync up my iPad app with the Zoom. And I tested it this morning and I made it work. And I think I'm going to manage to do it. So let us try right now. Okay. Oh, wait, I think I got to get out of, oh, there. So do y'all see it?
Yeah.
Okay, so this is this really cool little app that lets you play with lots of different things that we've been talking about. So in fact, I can let me annotate. I can show you the things here. So here is partial pressure of oxygen in your alveoli. Here's partial pressure of carbon dioxide in your alveoli. Frequency, this is how many breaths per minute. Here's the dead space. Normally it's about 150 mils. That's the dead space I was talking about. Tidal volume, this is what's the volume as you breathe in and out at a quiet breath. So I'm going to start by resetting this all. So here under normal quiet breathing, normally it's about 12 breaths per minute. Tidal volume is about a half a liter, 500 mils. And you can see the values of partial pressure of oxygen and partial pressure of carbon dioxide in the alveoli is what we've been talking about. 100 about for oxygen, 40 about for carbon dioxide. If I start breathing more quickly, here I am now breathing twice as fast, but just as deeply, you can see now I have a higher amount of oxygen, partial pressure, lower of carbon dioxide, which makes sense. If I am breathing deeper, now I'm having my breaths be twice as deep, 1 liter per breath instead. You can see I'm getting much higher oxygen and much lower carbon dioxide in my lungs. So by adjusting the frequency of your breathing and the volume, the depth of your breathing, you can change the levels of carbon dioxide and oxygen in your body pretty dramatically. When we get to your pH balance later, we're going to see that this actually is an important part of pH balance because that carbon dioxide gets converted to carbonic acid. So by having more or less carbon dioxide, you actually have lower or higher pH in your body. So you actually change your pulmonary ventilation frequency and depth to actually adjust your pH in the short term. So that's one of the things that's kind of cool to see here. The other thing, I'm going to reset now, kind of back to normal, but now I'm going to change the altitude. So here I have 760, that's just normal sea level where we're at now. Now we're going to Lake Tahoe, 600, and so 6,000 feet, it's about 609 millimeters of mercury. Now, just doing normal quiet breathing, you're only at 70 millimeters of mercury oxygen in your alveoli, which is not really enough to drive a nice diffusion of oxygen from your blood, from your alveoli into your blood. So all of a sudden, here I'm gonna increase, if I increase my tidal volume, increase my breathing frequency, I can bring it up. But I actually have to adjust. I have to breathe a little deeper and breathe a little more rapidly in order to make sure that I keep that partial pressure of oxygen up so I'm having an efficient transfer of oxygen into my bloodstream. Let's get up to Mount Whitney where it's 429 millimeters of mercury. So this is really high. This is, again, 14,500 feet. Right now, if you're not adjusting your breathing, your pulmonary, your oxygen in your lungs is below what is coming back from your tissues. You're actually having oxygen leave your blood in the lungs. So that's really not good. So you're going to have to really increase your tidal volume, increase your frequency, and you can bring it up. But even at your best, it's gonna be really hard to, feel like you're getting enough oxygen when you're up that high. If you've ever been hiking across the highest passes I've ever hiked at, I mean, just trying to like eat an apple is like, you know, you try to eat, and if you're just like eating an, if you're like trying to chewing an apple, and then it's like you're not just continually ventilating, you start panting, trying to catch up again after you've just tried to like take a bite of an apple. If we go up to Mount Everest, let's go to the top of Mount Everest, where you are at 29,000 feet. And again, the outside air has a pressure of only 226 millimeters of mercury. No matter how, well, this doesn't change. No matter how much you try, you cannot. You can breathe as fast as you can, as deep as you can. You can't get the partial pressure of oxygen high enough in your lungs to keep the air moving from your lungs into your bloodstream. So that's why if you try to go to the top of Mount Everest, you know, without an oxygen tank, you're gonna die. You can't keep your blood oxygen up there, because just the pressure of oxygen is too low, and that's what's driving the movement of the oxygen into your blood. That being said, if you're a bird, you know, we're not a comparative physiology class, but bird lungs are awesome. Bird lungs are really fun. They actually do have the ability to move oxygen into the blood, even when it's much lower oxygen outside, because they don't have all this dead space. And I'm not going to talk, we don't have enough time, but if you want to go read about bird lungs, they're really, really cool.